Okay, we're back with second 203 nerve 2. Well, let me set the slideshow. All right, I think we ended right here with pumps and channels. We talk about the sodium potassium pumps that help maintain the sodium potassium concentration gradients across the membrane, which are absolutely critical for nerve function to work. Okay. And they are going to be all over the cell. We'll be talking about these channels too, and the different channels are in different places. Pumps are all over the cell. In fact, they're all over every cell in your body, except maybe red blood cells. And a good fair amount of the ATP you use during the day is to work those pumps, because our nerve cells are leaky. They'll leak. If you look at these channels here, channels are really just holes in the membrane that let ions through. Remember, ions have a full charge. They're not permeable to the membrane. You have to have facilitate diffusion. You have to have a membrane protein to let an ion through. And these are called ion channels. Some of them are leakage channels. Why they exist, I don't know, but they leak. They let sodium leak into the cell and let potassium leak out. Remember, sodium is always more concentrated outside than inside. Potassium more concentrated inside than outside. And they leak. Well, this is the thing. The very first organism to have a nervous system was a similar thing to a jellyfish. They're called cnidarians or ctenophores. And they were really one of the very first multicellular organisms, multicellular animals. And the really first one to really have a nervous system. And the nerves that we had, the neurons that we have evolved from then, and that's about 1 billion years ago. That's when this first happened. Maybe 800 million. Anyway, and we evolved very similar nerve cells, and they're still leaky. They're still really leaky. They were leaky back then. Well, I guess maybe 500 million or maybe 600 million years ago. They were leaky back then. They're still leaky, and they work the same way. They pretty much work the same way as they did 500 million years ago. As a matter of fact, all animal nervous systems work the same way. And most of the research that has been done to find this basic neurophysiology was done using invertebrates, particularly sea slugs, because they have gigantic neurons. You can practically see the neurons with your naked eye, or at least you can see them under a microscope. You can't see human neurons under a microscope. They're way too small. Anyway, so channels are holes in the membrane that let ions pass through. And they're specific. There's sodium ion channels, potassium ion channels, calcium ion channels, chloride ion channels. That's the big four. And they only go through, well, it's a little bit of slop between sodium and calcium, because they're both cations and they're similar sizes. But usually ions just go through their own individual ion channels. And also, a lot of channels are gated. The leakage channels are not gated. What gated means is they have a door that is closed, and if it's closed, the ions don't go through the ion channel. If it's so it's a gate. It's a gate in the fence. So if the gate is closed, there's no ions traveling. The gate has to be opened for the ions to cross the membrane. And there's several ways you can open the gate, or the way the gates are controlled. One is chemically gated. That is, there is a there has to be, there's a receptor for a particular molecule associated with the voltage channel, the ion channel. And that molecule has to bind to the gate to open it. That's called chemically gated. Then there's also mechanically, I don't have mechanically gated down there. Some channels are open when you distort the membrane. And when you distort the membrane, that opens up the gate and ions channel. And then there's voltage gated. And that is, they're open when they're subjected to a particular voltage change. If you get that voltage change, they're open. At this voltage, they're open. At this voltage, they're closed. And usually, gated channels just have two states, open and closed. Except for sodium channels. They have three states, open, closed, and inactivated. And inactivated means they're closed. But what the difference is between closed and inactivated is a closed channel can be opened. An inactivated channel cannot be opened. Right? That's the difference. And only sodium channels can be inactivated like that. They have three states. Okay. We'll see how that makes we'll look at the importance of that later on when we get into the action potentials. Okay. So here are some, so here's the sodium potassium pump, or sometimes called the sodium potassium ATPase, because it has an enzyme associated with it that breaks ATP. And so what it does is that it gathers three sodiums, breaks open an ATP that provides the energy to push those sodiums out of the cell. And this is against the concentration gradient, remember, because sodium is always more concentrated outside than inside. So you're going in from an area of low concentration to an area of high concentration. So you have to use ATP to do that. And then it grabs two potassiums, pulls two potassiums into the cell, again against the concentration gradient, because potassium is more concentrated inside cells than outside. And that goes on 24/7 your whole life. All right. So it's also known as an electrogenic pump because these are cations, positively charged ions. And you're taking three positive charged ions and you're kicking them out. But you're only taking in two positive ions inside. So you're making the outside more positive and the inside more negative. And that's the situation all the time in the membrane potential. All of your cells are slightly negative inside with respect to the outside. Right? That's called the membrane potential, and we're going to be talking about that and explaining why that exists. And for nerve cells, it's minus 70 millivolts. That is 70 thousandths of a volt. So you're not going to shock yourself here. Very tiny potentials. And remember what a potential is. It's a separation of charge. Difference in charge from one place to the other is a potential. Because you can do work with that potential. And so all your cells are slightly negative with respect to the outside. Okay. Leakage channels. Here's a sodium leakage channel. So sodium just leaks in through these little holes in the membrane, constantly leaking in, following the concentration gradient by simple diffusion. Okay. Why are they there? I don't know, but they are. It's kind of crazy that they are, but they are. I guess the first, like I said, the first nerve cells that ever evolved had them, and you couldn't get rid of them. They're still here. Okay. Here's a chemically gated channel. So here's a little receptor for a particular molecule. This molecule, this little orange diamond. There's the gate that it's closed here. Okay, the molecule, and these are mostly neurotransmitters. This happens in synapses a lot. The neurotransmitter binds to the gate, opens the gate, allows the ions to cross the membrane. In this case, it's a potassium ion channel. So potassium now will move out of the cell into the synaptic cleft. Okay. Then voltage gated channels. It says 3 different states, only for sodium. Closed resting states. So you see these little pluses and negatives here. So this is the outside of the cell, this is the inside of the cell. And here this, and so we're at a, and so if it's negative inside, the sodium gate is closed. But we're gonna have a reversal of charge, and we're gonna briefly turn the inside of the cell positive. And how that happens, I'll let you know later. You have a reversal of charge, and now the inside is positive and the outside is negative. This will open the sodium ion channels, and sodium will allow and this will allow sodium ions to rush into the cell. But there's one particular situation where a cell is still positive inside and negative outside, and now this gate is inactivated. It cannot be opened up again, even though the inside of the cell is still positive. No ions can pass. It's inactivated, right? And it will only be it will only be able to be opened again when the inside of the cell becomes negative again. Right. Okay, distribution of pumps and channels. Where do you find them on the cell? And we're going to have to label parts of the cell also, and talk about segments of the cells, receiving segments and sending segments and so on, so transmissive segments. So both leakage channels are throughout. The pumps also throughout the entire cell. It has sodium potassium leakage channels and sodium potassium pumps. Potassium is more permeable because there are more potassium leak channels. Okay, so potassium is actually the most permeable ion. Functional segments of the cell. There's a receptive segments. This receives information from other cells or from other things that are happening on the outside. It receives information. These are usually dendrites, right? And they have usually chemically gated potassium, sodium, and chloride channels, or sometimes mechanically gated. Depends upon, when we get to the sense chapter, we'll learn about different senses, and chemically gated senses are smell and taste, but touch is mechanically gated. Okay, so that's the receptive segment. The initial segment is where integration occurs. This information that's being received is added together, and that's where the decision is made whether to turn the cell on or not. This is called threshold. And these, in the initial segment, sometimes called the axon hillock, also sometimes called the trigger zone, you have voltage-gated sodium and potassium channels. So they are open and closed. If the cell becomes less negative inside, this can open up these channels. Okay. The conductive segment. This is where an action potential is generated. And we're going to get to what an action potential is. It's the nerve signal. It's generated at the initial segment and is conducted down the conductive segment. And that's generally the axon, right? And those are voltage-gated channels as well. Transmissive segment, that's also called the axon terminals. They have calcium channels instead, voltage-gated calcium channels and calcium pumps. And this is where the information is going to be sent on to another nerve cell, a muscle or a gland or something. That's a transmissive signal. And this, oh, I didn't open the book again. This is right in the book. Shows you where the different segments of the cell are and what kind of pumps and channels that we have in them. Here's the receptive segments. These are the dendrites over here, and that's the cell body. Chemically gated cation channels, which are sodium and potassium channels. So chemically gated potassiums, chemically gated chloride, and chemically gated sodium. I don't know why they say chemically gated cation, because of sodium and potassium are both cations. Okay, there's the initial segment, also known as the axon hillock, as I mentioned. There we have the first voltage gated and potassium and sodium channels. So the cell body, that's part of receptive segment too. Dendrites and cell body, receptive segment, axon hillock, initial segment. Then conductive segment, we again have sodium and potassium voltage gated channels down the whole axon here to the axon terminals. And these are synapses down here. This is the transmissive segments, voltage gated calcium channels and calcium pumps, which will pump calcium from the inside to the out. Calcium is like sodium, it's always more concentrated outside than it is inside. And notice that these, over the entire neuron, we have the two leakage channels and a sodium potassium pump. Okay. So now we've talked about electrophysiology. And I already defined potentials. Potentials is a separation charge that some place has this charge and the other place has a different charge. So it's a separation charge. And it's a potential because it's a potential to do work. And that's just how a battery works. See, your cells are actually like little tiny batteries. You have a battery, the little nipple area on a battery is the positive pole. And then the little indentation on the other side of the battery is the negative pole. Right? This is a separation charge. And therefore, if you open up some kind of transmissive segment between those two things, charged particles will flow, and that's a electrical current. And electrical currents can do work. All right. So we're talking about a lot of different potentials. Like the resting membrane potential. Okay, this is, like I said, all of your cells, when I'm talking about nerve cells, I have a resting membrane potential of minus 70 millivolts. So there's slightly more negative inside than outside. And we're going to talk about, so there's a disequilibrium of ion concentrations across the membrane. It means that there's concentration gradients for ions across the membrane. As I mentioned, sodium always more concentrated outside than inside. Potassium always more concentrated inside than outside. Chloride is also always more concentrated outside than inside. And calcium is more concentrated outside than inside. The only one in the opposite direction is potassium. Okay, so we're going to talk about concentration and electrical gradients. That is an electrochemical gradient across the membrane and how it gets established. And that's what I'm going to talk about right now. Okay. I'm doing something a little different. It's really hard to describe this in words. And so I usually draw it on the board. And so I'm going to attempt this now. Come a little closer here. And I'm going to kill the screen share for a second. So we get the full screen. And I have here a cell membrane right here. All right, let me get the marker. Now let's make believe that there's a cell. So here's the inside of the cell. Here's the outside cell. And let's just make believe there's nothing on the outside of the cell. No ions or anything on the outside. But the inside, there are potassium ions, and there are big proteins that are usually negatively charged, all right? So there are, and now we're going to put in some potassium ion channels. Here's a potassium ion channel right here. All right, so so potassium is permeable now, and potassium will leave the membrane by simple diffusion. We got a concentration gradient, and more concentrated in here than out here. All right, so potassium will leave the cell and bring positive charges outside. Protein, not permeable. It's not going anywhere. So as more and more potassiums leave the cell, more and more positive charges build up here. And what we got left over here is negative charges. So it becomes negative on the inside and positive on the outside. This potassium is leaving and bringing positive charge out and leaving the negatives behind. That's the concentration gradient, all right? The chemical gradient. But now, as that goes on, we have established an electrical gradient. Well, what do we know about opposite charges? They attract each other. So since the inside becomes more and more negative, and potassium is positively charged, potassium is not going to be attracted to the inside of the cell. So there's going to be potassium channels where potassium is going to come back in because it's positively charged, and the inside of the cell is negative, and opposite charges attract. Right? So that'll happen, right? That's the electrical chemical gradient. Two forces at work here. Chemical gradient pulling potassium out of the cell. Electrical gradient pulling potassium into the cell. All right. Okay, that'll go on until we reach an equilibrium, where for every potassium that leaves the cell, one potassium enters the cell. So the potential doesn't change. And this is called, this is an equilibrium of a sort, and that's why it's called the equilibrium potential. And that is at minus 90 millivolts. It's what's called the potassium equilibrium potential. It's a really good thing to understand this and get this, because for some reason it's not in the book. It really should be in the book, but it's not. We should talk about Ms. Bido about that, putting in, because it's really important. At minus 90 millivolts, the concentration gradient is exactly equal and opposite to the electrical gradient. And this will never change. Okay. But neurons aren't at minus 90 millivolts. They're at minus 70 millivolts. Which means the concentration gradient is slightly larger than the electrical gradient. So more potassium will leave than enter. Why is it minus 70? Why is it not minus 90? Because I've left out the other ions. We got sodium ions. They're out here, and they really want to come in. Because for potassium, the concentration gradient and the electrical gradient are in opposite directions. Right? For sodium, they're both in the same direction. Sodium wants to come in by concentration gradient. It's more concentrated out here than in here. It also wants to come in because of the electrical gradient. It's negative inside and it's positive. So it really wants to come in. And its equilibrium potential, remember every ion has an equilibrium potential. Its equilibrium potential is plus 60, way up there. So if we got the inside of the cell to be plus 60, the concentration gradient and electrical gradient for sodium would be equal, and for every sodium that came in, a sodium would leave, and it would never change. Okay. And then we have, remember, we have calcium out here and chloride out here, and they all are equilibrium potentials also. So the minus 70 millivolts is an average of all of the ions' equilibrium potentials, all the equilibrium potentials of each ion. And it's closest to the potassium equilibrium potential, because we know that potassium is the most permeable ion. Right? So it has the major role in setting the membrane potential. Okay. So what we have here, oh yeah. So you have to go into a little basic electricity, which I was going to put up on the next slide. We're talking about Ohm's law, V equals IR. There's three things in electricity: voltage, current, and resistance. Voltage is the size of the electrical gradient. Current is moving charges, and resistance is how hard it is for these charges to move. Right? So remember, here's a battery here. Like I said, a little nipple here, and then a little indentation here and a battery. And so the little nipple is positive. The indentation is negative. So when you connect the wire from here to here, let's say there's a little switch in the wire here. Connect the positive and negative pole with a wire. And then you close the gap here. And so now, well, in electricity, in like house current, it's negative charges that move. Electrons move from the negative pole to the positive pole. And that's an electrical current. And what can you do with a current? You do work. You have a resistance in here. And this could be a little motor, could be a light, could be whatever. And we do work using this electrical current. Well, let's make that a cell membrane. The same thing, separation of charges. And you open up an ion channel, and positive, you have charged particles moving. In the case of bioelectricity, it's ions, not electrons. And you can do work with that. Right? So all of our neurons are little tiny batteries. Think of it like that. All right. So let's go back to screen sharing. Oh, wait, it's this one. Okay, oops. So as I mentioned, there's three things in electricity: voltage, current, and resistance. And They're related to each other. Hey, what happened to, wait a minute, go back. So I didn't write down Ohm's law. V equals IR. Is Ohm's law. And it's in different ways you can divide both sides by R. You can see current is equal to voltage over the resistance. So the greater the voltage, the greater current. The greater the resistance, the less the current. Okay, so charged particles. So current is the movement of charged particles. Charged particles in electrophysiology are ions. There's a difference in charge distribution across the membrane. Inside the cell, slightly negative with respect to the outside. The membrane offers resistance to charged particles. That's the R. Resistance is altered by opening and closing ion channels. So if the ion channels are closed, the resistance is maximum. Ion channel is open, resistance is minimal. And current is generated when ions are allowed to cross the membrane. And again, you can use that current to do work. So as I mentioned, the resonant potential is usually minus 70 millivolts. For nerve cells, it's usually minus 70 millivolts. It's different in some other cells, but in nerve cells, minus 70 millivolts. And the role of potassium, I just described, is the most important ion in setting that resting membrane potential because it's the most permeable. And the role of sodium and other ions are to modify the inside from the potassium equilibrium potential to minus 70. So like I said, it's like an average of all the equilibrium potentials added together gives you minus 70. It's closest to potassium equilibrium potential because potassium is most permeable. So it's really far away from the sodium equilibrium potential because sodium is not nearly as permeable as potassium is. Okay, and the role of the sodium-potassium pump, as I already mentioned, it's an electrogenic pump. It helps maintain the inside of the cell to be negative because it pumps 3 positives out, but only pumps 2 positives in. So it makes the inside of the cell more negative. So it helps to maintain the membrane potential. And changing the rest of the membrane potential. We are going to change the rest of the membrane potential, and that's the basis behind nerve signals, is that if we make the inside of the cell more positive, by letting positive ion in, that's called depolarization. And that usually means the cell is excited, which means it gets closer to sending a nerve signal, an action potential. If you make the inside more negative, that's called hyperpolarization. And that's usually inhibition that keeps the cell off and makes it less likely that it's going to turn on. Okay, so potentials. Equilibrium potential when the forces are equal. When a chemical force is equal to the electrical force. Local potentials are potentials that are generated usually by chemically gated ion channels, and they usually happen in the receptive segment, the cell body, and the dendrites. And they are graded, which means they're different sizes. They're decremental, which means the farther they travel from where they are initiated, the smaller they get. They're reversible. They can go either direction. And it can be excitatory, which makes the cell more likely to turn on. Or they can be inhibitory, and make the cell less likely to be turned on. Generated by the action of membrane channels controlled by chemicals, light, or mechanical distortion of the membrane. So yeah, that's right. Light is something that can affect whether ion channels are open or closed. And of course, that's the only place that has an effect is in vision, and we'll get to that in the sensors chapter. Then there's what's called an action potential. This is like completely the reverse of local potentials. For one, it's all or none. It's either this big or nothing. It's not graded. There's no little bit and little larger. It's this or nothing. It's non-decremental. Doesn't matter how far it travels, it's always the same size. It's also known as stereotypical. It never changes, it's always the same. It's irreversible. It goes in one direction. That's it. It doesn't go in two directions. For reasons we'll get to. And like I said, it's unchangeable or stereotypical. Every exponential is exactly like every other action potential. Okay. And it's generated by the activation of voltage-gated membrane channels at the axon hillock. Right? Okay, there are the segments again of the cell, the receptive segment, binding neurotransmitter, release from presynaptic neuron, production of gradient potentials. In the cell body and the dendrites. Initial segment, summation of gradient potentials, initiation of action potentials. So these gradient potentials are local potentials, same thing. Are added together, and if you depolarize the cell to a certain extent called threshold, you will fire an action potential. And that will be propagated down the conductive segment, the axon, to the transmissive segment. And an action potential will cause release of neurotransmitter that will activate another cell downstream, either another nerve cell, a muscle cell, a gland cell. Okay, let's we'll talk more about this. Let's call it because it's been almost 40 minutes. And so let's turn this on, and I'll pick it up at this slide in the next video lecture. So remember, this is the 203nerve2. So I'll see you in the next lecture.